Welcome to the next episode of the Worm Podcast.
It is fantastic to be back diving in.
Before we get started, just a quick announcement. The UKC Elegans Meeting 2026 is happening from the 15th to the 17th of April in Southampton.
Oh, perfect.
Yeah, it's being hosted by Vincent O'Connor and Lindy Holdendye at Southampton.
Which is actually a perfect segue because they are also some of the authors on the paper we're talking about today.
Exactly.
Alongside Eleonora, Maria Pironi, Pamela Imperador, Graziano Fiorito, and James Dillon. A really great team.
Absolutely. So registration and abstract submission details are in the show notes. We'd strongly encourage you to attend. Now on to this week's deep dive.
Right, so today we're looking at a paper from Biology Open, published in 2025, and it involves a really fascinating mix of creatures.
It's an unusual pairing.
It is. We are bridging the gap between the common octopus, Octopus vulgaris, and our favorite nematode worm, Caenorhabditis elegans.
The meeting of the minds? Or maybe the meeting of the tentacles and the microscopic wigglers?
That's about right. And the whole mission of this deep dive is to understand how scientists can study pain, or to be technical, nociception, in an animal that's just notoriously difficult to work with.
The octopus.
The octopus. And they do it by using this model hopping technique with a much, much simpler animal.
The worm. I love that term, model hopping. It sounds like some kind of scientific sport. But okay, before we get into the hopping part, why do we care so much about octopus pain in the first place?
Well, this really gets at a major issue in modern biology. I mean, octopuses are incredibly intelligent. They're complex.
They solve puzzles. They use tools.
Distinct personalities, even. And because of that complexity, they're protected under these really strict welfare laws. Directive 2010-63 EU. Specifically.
Right. So legally, we're already saying we believe they can feel pain.
We are. But here's the problem. Scientifically, it's a bit of a black box. We assume they feel pain from their behavior, but we don't actually know the molecular machinery, the biological hardware inside.
So we're protecting them based on how they act, but we don't know what's going on under the hood.
Exactly. And that is what this paper calls the octopus problem.
Yeah.
And you might ask, why not just study the octopus directly?
I'm guessing they're a little high maintenance.
That is, putting it mildly. The paper goes into some of the logistical nightmares. First, you have husbandry issues.
Just keeping them alive in a lab.
It's incredibly hard. They're difficult to breed. They're babies, the paralarvae, they're so delicate. They're picky eaters. The environment has to be perfect.
So you can't just order a batch of standardized lab octopuses like you can with mice.
No, not at all. Which leads to the second problem, sourcing. Most research octopuses are wild caught.
Oh, that's a nightmare for a scientist. You want clean data, right?
It's terrible for data. If you catch an octopus from the ocean, you have no idea how old it is. You don't know its genetic background. You don't know what kind of stress it's been through. All of that just introduces so much noise.
And I'm assuming we can't just pop in and edit their genes?
Not easily. We just don't have those sophisticated genetic switches for octopuses. In a fruit fly or a worm, we can knock out a gene. We can just delete it and see what happens.
If you delete a gene and the animal stops feeling heat, you've found the heat sensor.
Precisely. We can't do that with an octopus. Not yet.
So we have this smart, protected animal that we can't easily study to actually verify why it needs that protection.
That's the problem in a nutshell. And that is where bioinformatic model hopping comes in.
Okay, let's unpack this. How on earth do you use a worm to solve an octopus problem?
It's a really clever three-step process. Step one is just good old-fashioned data mining. The researchers started with a laundry list of known pain genes from animals we do understand really well.
Humans, mice, fruit flies, round up the usual suspects.
Exactly. They started with about 474 genes and then filtered that list down to 74 high-priority candidates, the ones that are really conserved across different species.
Okay, so they have their list of most wanted pain genes. Then what?
Step two. They look into the Octopus vulgaris transcriptome.
Right, the transcriptome. That's the library of RNA, the genetic instructions the octopus is actually using at that moment.
That's it. So they searched that library to see if the octopus has versions of these 74 genes. And they did. They found 51 candidate genes in the octopus that look like they're involved in nociception.
So the octopus has the parts. But just because you have a part doesn't mean you know how it works.
Precisely. Just because a gene looks like a pain sensor doesn't mean it acts like one. And since we can't delete the gene in the octopus to test it,
We hop to the worm?
we hop to the worm. They find the equivalent genes The orthologues and elegans.
I think we talked about an analogy for this. It's like finding a mysterious part in a Ferrari engine.
The octopus is the Ferrari, yes.
And you can't take the Ferrari apart. It's too complex, too valuable. But then you realize that same mysterious part is also in your lawnmower.
The worm.
The worm. So you take the part out of the lawnmower, and if the engine stops working, you know that part is essential.
And you can infer it's probably doing the same critical job back in the Ferrari.
So they took the octopus gene's cousin from the worm and started breaking it.
They did. They ended up testing 38 of these candidate genes using mutant worm strains.
Let's get into the actual experiment. You called it the drop assay.
It's a classic in the field. It's very simple, very elegant. You have a worm. It's just moving forward on a plate. You take a tiny drop of acidic solution.
What kind of acid?
Acetic acid at pH 3.
So basically strong vinegar.
Very strong vinegar.
Right.
Now, a normal worm, what we call a wild type, it touches that acid, it realizes it's noxious, and it immediately reverses.
Hits the brakes and gets out of there.
It does at least 3 full-body reversals in 5 seconds. It's a very clear, very robust avoidance behavior.
And why acid? Why not, say, heat or a physical poke?
Well, acid is just a universal ouch signal in nature. Protons cause damage to cells. And we already have data from other studies showing that octopus arms will pull away from acetic acid. They even groom the spot afterwards.
Which suggests it's more than just a reflex. It's like they're processing, ow, that hurt.
That's the thinking. So the logic for the worm is simple. If it doesn't reverse when it hits the acid.
Then the gene you broke was essential for feeling that particular kind of pain.
You've got it.
All right, let's get to the results. They tested 45 different worm strains with mutations. Which genes actually mattered? What were the hot sensors?
The heavy hitters, not surprisingly, were the TRP channels, transient receptor potential channels.
These things come up all the time in sensory biology, don't they?
They're the primary sensors of the world around us. So the study highlighted 2 main types. First was TRPV, the vanilloid type. In the worm, these are the genes OSM9 and OCAR2.
And when those were mutated, what did the worms do?
They barely reacted, just plowed right through the acid drop.
And for context, TRPV is famous in humans for something else, right?
It is. TRPV1 is our capsaicin receptor.
The thing that detects chili peppers.
Yes, it's what makes a habanero feel hot. It also detects actual physical scalding heat. So finding this whole pathway, conserved in the octopus, and then confirming it works in the worm, it tells you that the basic hardware for sensing a chemical burn is ancient.
It's like evolution came up with a smoke detector 500 million years ago and just kept installing it in every new building.
That's a fantastic way to put it. They also found TRPA channels, that's the tractable gene in the worm. That one often senses chemical irritants, like the sharp sting of wasabi or mustard oil.
So the big picture here is that the octopus likely feels the burn of acid using very similar machinery to us and to the worms.
That's the strong implication. But, and this is where it gets really interesting, they didn't just look for the pain sensors, they looked for the pain relief system.
You mean like natural painkillers?
The opioid system, exactly. Invertebrates, so us, mice, dogs, we rely on opioid receptors to manage and dampen pain signals.
Morphine, endorphins, all that.
Right. So the researchers looked for the genes for these receptors, mu, kappa, and delta, in the octopus transcriptome. They're not there.
Wait, the octopus doesn't have opioid receptors.
The octopus vulgaris transcriptome appears to be completely missing the standard vertebrate opioid receptors.
So, does that mean they can't feel relief from pain? That sounds horrifying.
No, not necessarily. It just means they don't use our system. It means they evolved a different system.
Okay, so what's the alternative they found?
They found genes related to a couple of other neuropeptide systems, elatostatin C, or AST-C, and crucially, the fMRFamide system.
fMRFamide.
It's a bit of a mouthful, but it's a huge family of signaling molecules in invertebrates. The researchers thought this might be the octopus's version of our opioid system.
So, back to the lawnmower.
Back to the worm. They took a worm mutant that was missing the fMRFamide receptor, a gene called FROPR3.
And what happened when they put the acid drop in front of that worm?
It stopped reacting. It failed to avoid the noxious stimulus.
Hold on, let me get this straight. If you break the pain relief system, shouldn't the worm feel more pain? Shouldn't it, overreact?
That is a very logical question. But biology is rarely that simple. In this case, it suggests that fMRFamide signaling isn't just a simple dampener. It's a critical part of the entire pain processing pathway. It might be modulating the signal, shaping it so it could be properly interpreted if you break that part of the circuit.
The whole system just crashes. The message doesn't get through properly.
Exactly.
Yeah.
And finding that fMRFamide is this central to nociception in invertebrates is a huge discovery. This really could be the octopus's opioid system.
Which has massive implications for how we treat these animals, doesn't it?
Oh, huge. I mean, think about it. If you have an octopus in a lab that needs surgery, you can't just give it morphine.
Because there's no log for that key.
Right. It might do absolutely nothing. You would need to develop drugs that specifically target their fMRFamide system.
That's incredible. We have to design alien medicine for an animal from our own planet?
Precisely. And the study didn't even stop at the receptors. They also looked at the manufacturing crew.
The enzymes that build these things.
Yes. You see, neuropeptides like fMRFamide, they don't just appear. They're built as these long, inactive protein chains, and they need to be chopped up into their final active forms.
Like cutting a key from a metal blank.
Perfect analogy. The study looked at proprotein convertases and carboxypeptidase E. In worms, those are the genes AGL3 and AGL21.
And let me guess, when those enzymes were missing.
The worms didn't react to the acid.
So even if you have the sensor and you have the receptor, if you don't have the enzyme to actually cut the key, the whole system fails.
The system fails. It proves that the processing of these neuropeptides is just as vital as the receptors themselves. And critically, the octopus has the genes for these exact enzymes.
It just shows how many different points of control there are. It's not one single pain gene. It's this whole complex network.
A network with some real surprises, too.
Oh, what stood out to you as a surprise?
The ASICs. The acid-sensing ion channels.
With a name like that, you'd think they would be the stars of an acid test.
You would. But what they found was that most of the single worm mutants for these ASIC genes, they reacted totally normally. They sensed the acid just fine.
That is strange.
It is. But then they tested a specific double mutant where two genes, AGD1 and degree1, were both broken. And those worms finally became unresponsive.
Okay, so why is that combination so important?
Because the degree1 gene in worms is known to function in glial cells, not just in neurons.
Glial cells. Those are the support cells, right? The ones we used to think were just like packing peanuts for the brain.
That's the old view. We now know they are active players in sensing and modulating signals. And the octopus arm is absolutely packed with glial-like cells.
So this is a hint that pain detection isn't just about the nerve firing. The support cells might be involved in sensing the environment, too.
It opens up a huge new area for research. If we're only looking at neurons, we could be missing half the story.
And there was one more channel type mentioned, wasn't there? Potassium channels.
Yes, the KCNQ channels, the gene KQT3 in worms. These channels basically regulate how electrically excitable a neuron is. And mutants for these also fail the acid test.
So a neuron's general readiness to fire is like a master volume knob for the whole pain signal.
That's a good way to see it.
So, okay, let's step back and look at the scoreboard. How did this model hopping strategy actually perform?
It was incredibly successful. Out of the 38 candidates they tested in the worm, they confirmed that 19 of them were directly related to nociception.
That's a 50% hit rate.
That is an amazing hit rate for this kind of exploratory genetics. They have effectively given the entire scientific community a priority list, a treasure map. Instead of guessing which of the thousands of octopus genes to study, researchers now have 19 high-value targets.
So bringing it all together, what does this all mean? Why should you, listening at home, care about a worm swimming in vinegar?
Well, if we connect all the dots, there are really three huge takeaways. The first, and maybe the most important, is welfare.
This validates the laws that protect them.
It provides the molecular proof. It's not just that we see an octopus pull its arm away. We now know it has the specific components, the TRPs, the peptide systems, the enzymes, that are required to detect and process a painful stimulus. It moves the conversation from, we think they feel pain, to, here is the biological machinery for pain.
It's hard evidence. What's the second takeaway?
It's the evolutionary perspective.
Yeah.
It's beautiful, really. It shows that even though the eyes and brains of a human, an octopus, and a worm look completely different, the basic chemical language of ouch is shared across 500 million years of evolution.
We're all using the same fundamental toolkit.
We really are. Protons, TRP channels, peptides. This system is ancient. We are far more connected to that little worm than we might like to think.
Fair enough. And the third takeaway?
It validates model hopping as a research strategy. I mean, think of all the other fascinating but intractable animals out there, deep-sea squid, exotic insects. This paper provides a roadmap. It proves we can use the tractable to understand the intractable.
Use the lawnmower to figure out the Ferrari. So the researchers have essentially drawn the map for the next decade of cephalopod pain research.
They really have. And they've highlighted that we need to stop looking for purely human systems in other animals. If we had just stopped when we saw no opioid receptors, we might have made the wrong conclusion. But by digging deeper, we found their alternative, the fMorphamide system.
Which brings me to a final thought to leave you with. If octopuses lack our opioid receptors, which are tied to feelings of relief and even euphoria in us, but they have this completely different fMorphamide system, does their version of relief feel totally alien to ours?
That is the million-dollar question. What is the subjective experience of a creature with such a profoundly different neurochemistry?
And on a practical level, if we want to be ethical in how we treat these animals, we can't just give them our medicine. We have to design painkillers that target their specific biology. We have to create medicine for an alien physiology that evolved right here on Earth.
It forces us to step way outside our comfortable vertebrate-centric view of the world.
Absolutely. Well, that brings us to the end of this deep dive. A huge thank you to all the authors, Pironi, O'Connor, Holdendye, Imperador, Furito, and Dillon for this incredible piece of work.
It was a true pleasure to unpack it.
And one last reminder, the UKC Elegans Meeting 2026 is in Southampton, April 15th to 17th. If this kind of science excites you, that is the place to be.
Definitely check it out.
Until next time, keep diving deep.
Goodbye, everyone.